Okay, so in this video, I'm going to talk about the osmosis lab experiment that we'll be conducting. So before coming to lab and performing the experiment, you should read through the lab packet and also complete the pre-lab questions. Reading through the lab packet, especially the protocol, will help you to prepare yourself. There is also a lot of information about the plasma membrane and about osmosis in both chapters 4 and really in chapter 5. And so your textbook is a great reference to refer to for learning about the structure of the plasma membrane, what it's made of, diffusion, passive transport, and osmosis. So all of that terminology is right in your textbook. So again, that's a great source. I'm going to make a second video talking about how to find primary sources for this paper as well. But first, what I wanted to do is to talk about the data and to talk about kind of how you're going to gather this data and what you're going to do with it. And so again, if you've read the packet, the procedure tells you that you're going to make 9 cells total, okay? And so 3 cells will be in group A, 3 cells will belong to group B, and 3 cells will belong to group C. And so the cells will be made by adding 15 milliliters of volume of either water or the 60% sucrose solution. And then the cells will be placed into beakers with, again, 200 milliliters of either water or the 60% sucrose solution. And so what you have to do is prior to putting your cells into the beakers, you have to weigh them. And so again, there's a table in that packet where you will record the weight of each cell at time 0. So that's the starting weight, the initial weight. Then you're going to drop each cell into its corresponding beaker and let it sit there for 60 minutes. And so during that 60 minutes, you can really start your lab report. Okay, so make sure you either bring your computer from home, or again, there's 4 computers in the lab, 1 per group. You can start to look up reference articles, primary literature, and you can even start to build your own lab report. Okay. At the end of 60 minutes, you're going to pull the cells out of their corresponding beakers. You're going to, kind of dry them off. So pat them dry with a paper towel, and then you're going to reweigh your cells. And so that'll be the weight of your cells at 60 minutes. And then what's going to happen is we're going to share data as a class. Okay. And so if there are 4 groups, right, and each group does 3 cells for each scenario, so each group, you should have a total of 12 cells for group A, B, and C. So you should have before and after data, 0-minute and 60-minute data for 12 cells. This is also nice because sharing data as a class means that we should all have the same data. Okay, so all of you, your graph should look, relatively the same. Your averages, your standard deviation, your statistical tests should all be the same. So when we look at each other's lab reports in a week from now, you will be able to really tell if your data makes sense, if you did the calculations right. Okay, so that's important to note too. So your job between this week's lab and next week's lab is to write a rough draft. Okay, so all of the parts and pieces of the lab report, you need to put in this rough draft. You should follow the guidelines that are in the packet, and you should come in with a paper that you believe is good enough to hand in. That's what your rough draft should be. And during next week's lab, we're going to kind of review each other's rough drafts, look at the data, make sure everything looks good, and then you have 1 more week to make your report hopefully perfect. Okay. You should bring a hard copy of your lab report with you next week so that you have something to share with your peers. Don't bring in, an electronic copy. It needs to be a hard copy. Okay. All right. So I've got on our screen here an Excel spreadsheet of some made up data. Okay. So these are not the numbers that you're going to get, but I wanted to do a little bit of practice with you. And so what I have here, again, are group A, B, and C, and I have before and after data for all of the cells. So again, the cells are going to be weighed before. You'll have 3 for each category, but you're going to share data as a class. And so you can see up here that I think I have 12. Maybe I only have 11 cells. Well, whatever. You'll have 12 cells for each of them. I think I might only have 10. You'll have 12. Okay. And so the first thing you're going to do is you're going to calculate the mean for time 0 and time 60 minutes for each set of cells. Okay, so in my first column here, I've got the group A before data. So again, these are those cells at time 0. And I've got down here a row for the averages. And so in my first cell of that row, I'm going to write equals. All right, so again, if I put an equal sign, that tells Excel I'm ready to put a formula. So Excel, you need to calculate something for me. And I'm going to start to type in average. And the first one that pops up is average. I'm going to double click on that. And so now Excel says, sure, I'll calculate the average of what? And so I'm just going to go up here to the before, group A before or time 0, and I'm going to click on that first cell's weight, and I'm going to drag to highlight all the cells in that column. Okay, so cells B3 through B12, I want the average of those. So again, I'm going to do end of parentheses, and I'm going to hit enter. And so the average of my first groups before is 15.34. I'm going to do the same thing for my 60 minutes. So this is after the 60-minute time interval. Let's see, equals average. So again, double clicked, highlight the data, end of parentheses, and equals. Okay, so my average before is 15.34. My average after is 15.19. Not a huge difference. Okay, so that cell didn't really gain or lose weight. Let's go to group B. So again, equals average. And I'm going to highlight my data for my before, end of parentheses, and equals. Okay, so my cell here weighed just a little bit more than my group A cells. My average weight is 16.15, and my average weight after, I don't know yet, let's see. The data, end of parentheses, enter. So this set of cells looks like it lost a pretty significant amount of weight. We'll see if it's actually significant in just a minute. For group C, so same thing, equals average. So you really got to know the formula for average. Highlight the data I want it to take the average of. There we go. There's the mean value for group C. And then equals average. So again, you should be proficient at calculating the mean or the average for data. It looks like that group of cells gained some weight. Okay, so again, this is made up data. Your data may not correspond exactly to this. Don't worry if it doesn't. It may not. Okay, so these are all made up numbers. So now I'm going to do the standard deviation for each of these. Okay, so again, equals STDEV, and you want to do the sample one, so STDEV.S, so the second one down on my list. And now again, I just highlight the data. I want to know, okay, what is the standard deviation for this group of numbers? Okay, so the deviation is 0.45 about for my time 0 for group A equals STDEV. So again, there's group my second set of data. I do my third here, equals STDEV. Standard deviation there. Next one, equals. If you forget the equal sign, it won't calculate anything for you. Okay. And so again, equals STDEVS. Parentheses equals, and my last one, equals STDEV. Go and highlight my data. Okay. So now I've got my mean values and I've got my standard deviation. I'm going to now make a graph, but before I do that, I think I'm going to show you quickly how to run the last statistical test. So again, there's two ways that I can do this. I can use the data analysis toolpack, or I can just plug in a formula here to calculate p-value. And so we're going to, in this case, we are going to compare before and after data to figure out if there is a statistically significant difference. So you actually have to run three t-tests here. You're going to run one t-test for group A, and again, you're asking, is there a difference in the weight of these cells before and after? You'll ask the same question for group B. Is there a statistically significant difference in the weight of the cells before versus after? And the same thing for group C. Is there a statistically significant difference in the weight of these cells before and after? So in your hypothesis section of your introduction, make sure that your null and alternative hypothesis have to do with that, right? So we're testing to see if there is a difference in the weight of these cells before and after. Okay, and just by looking at it for group A, these numbers look really close, 15.34 and 15.19. So my guess is that there's not a statistically significant difference. But for groups B and C, it looks like there might be. So again, we need to do the calculation to make sure. And so let's do that. So here we go, equals T dot test. Okay, so again, without using the data analysis tool pack, which you could use, or you could just use this formula. Okay, so there, this pops up and it says, okay, I'll do a t-test. Of what? So it says array 1. So this is saying, give me the first set of data. And so I want a t-test done on this data, this data. So this is the data that I'm comparing in my first t-test, before and after data for group A. Okay, so again, I selected the first set of data, I put a comma, and then I selected the second set of data. I'm going to put another comma, and this dialog box is going to pop up, and you're going to select two-tailed distribution, and you're going to put comma again, and now it's saying, okay, what type of t-test do you want to do? So for the first two labs that we did, we were using a two-sample unequal variance, but this time we're looking at before versus after data. And so this is called paired data. And so we're going to use a paired t-test, which is the first one. So I'm just going to double click on that. I'm going to do an end of parentheses, and I'm going to hit an enter. Okay, and so my p-value for this first set of data is 0.4 to 4. Okay, just to round up. So what does that mean? This is the important part. So if you have a p-value that is greater than 0.05, that's the golden number that you got to remember, 0.05. So if your p-value is greater than 0.05, then there is no statistically significant difference. And so in this case, the null hypothesis for group A would be supported. There is not a difference. Okay, why? So look at your p-value. The p-value that we just calculated is 0.4, which is greater than 0.05. Okay, so there is no statistically significant difference. Let's go to our second set of data here. Okay, so I'm going to go under group B. And again, I'm going to calculate the p-value for this before and after data. And so equals, and I'm going to do T dot test. So again, that's the formula. I'm going to double click on that. Same dialog box comes up. It says, okay, what data? So what's your first set of data? My group B before, numbers, comma. My second set of data is here, my after data, comma, two-tail, comma, paired. End of parentheses equals. Okay, now let's look at this number here in this cell. It says 7.008, blah, blah, blah. And then it says e to the negative 12th. Okay, so be careful here. You might look at this and say, oh, it's 7. Oh, that's way more than 0.05. That's not the number, okay? This is an exponent, so e to the negative 12th means that you have to take that decimal place and move it to the left 12 spaces. So it's actually a very, very small number. It's 0.0000007. So again, 12 places out. So it's a very, very small number. So just as a reminder here, your p-value is going to be between 0 and 1. You're never going to have a p-value over 1. It's between 0 and 1. Okay? And so this number is very, very small. It is less than 0.05, much less than 0.05. And so you can say with confidence that there is a statistically significant difference in the before and after data for group B. And so this loss of weight for group B is statistically significant. Okay, let's go to group C. So again, equals T dot test, double click, select the data. So here's the before data, comma, here's the after data, whoop, comma, two-tailed, comma, paired, end of parentheses, equals. Okay, so same thing, look at this, it's 4 point something something, e to the negative 8th. Okay, so again, that decimal place next to the 4 needs to be brought to the left 8 times. Okay, so again, this is a very small number, much smaller than 0.05. So again, this provides evidence that there is a statistically significant difference between the before and after for group C. Okay, so again, this is a very fast way to run a t-test. And again, for this particular experiment, you're going to do a paired t-test. And that's because we're dealing with before and after data. So it's data that's paired. Okay. The one last thing I want to show you is how to make a graph.